Thank you again for this invitation. It's nice to be able to connect with so many folks from across the country. So I'm going to be talking about an invasive species that we have been dealing with here in Kearneysville, West Virginia, and throughout the Mid-Atlantic, the spotted lanternfly, and specifically talking about what we've learned about its host range, as well as their potential spread through human-assisted transport. So spotted lanternfly, or SLF as I'll refer to it, is an invasive species in the US, South Korea, and Japan. Its native range is parts of China, India, and Vietnam, and this is a typical fulguroid. And it's been in the US for a number of years, and so we'll get to that. In terms of its life history, it's a little bit different than some of the other invasives that I've worked with throughout my career in that it has a single generation per year. It overwinters in the egg stage, which is kind of just this nondescript bit of eggs hidden under what looks like kind of a bit of mud. In the spring, eggs hatch, and there are four nymphal instars. The first three instars are black with white spots. The fourth instar is red, very apparent, with black and white spots. And then the adult is a very colorful adult, and it feeds for quite a bit of time. In fact, from about July till September here in the Mid-Atlantic region until it reaches the adult stage. And then they start laying eggs again. Just about now, we're starting to see oviposition in the field. Its current known distribution in the US, it has now, it originally was first detected in and around the Reading, Pennsylvania area where you see the red star. And it's thought that eggs were transported inadvertently on a shipment of stone. And from there, and that was back in 2014. So from there, you can see the continued spread. It's now been detected in 17 states, throughout the East, and this is at a county level that really shows where lanternfly is present. It's also still a regulated pest, and so there are a number of campaigns at various states to try to reduce the spread of this insect from this region. Now, I wanted to show you what it looks like after you've had spotted lanternfly present for a number of years. These photos and this video were taken just over the last week at our facility. This is our water tower, and all of that stuff you see at the bottom are spotted lanternflies, adults. At this point in the season, the adults orient to dark upright objects, essentially tree trunks, looking for a place to feed. These are typical phloem feeders, so they're going to feed through the bark of trees. So this looks like a giant tree if you're a spotted lanternfly. But the question, of course, is how do you go from just a couple of invaders to just exponential growth like you're seeing here? There are a number of hypotheses that are typically put forward when we talk about invasive species. Things like the enemy release hypothesis, meaning that the insect is separated from its co-evolved predators, parasitoids, and pathogens. Another is that the invasive or exotic species can outcompete native species for resources. The fact that they are strong dispersers, maybe they can move through the environment and invade new regions, allowing them to reach these resources and establish and grow in terms of the size of the population. Also, if they land in a place that has favorable climatic conditions, if they have a high reproductive rate, and if they have a generalist diet, meaning they'll feed on lots of stuff. And so in this case, what I'm going to talk to you all about is some of our work associated with this generalist diet and also the idea of their dispersal and how that may have contributed to what we're seeing now, which is some pretty big populations and some pretty big issues. So in terms of generalist diets, when spotted lanternfly first invaded Pennsylvania, eastern Pennsylvania in 2014, we didn't know much. And there were some papers that came out over the past few years to look at what was known in sort of the published literature, including the fact that based on their native range in Asia, parts of Asia, that they were known to feed on 103 different plant taxa from 33 families, that they principally fed on forest tree species. Also, that tree of heaven was a key host for them, Alanthus altissima, and that in more recent studies that they can complete their development in the absence of this host, but fitness was reduced. And then the fact that we see this broad host range in the early stages of their development, early instars, nymphs, but it becomes more narrow as the insect continues to complete development to the adult stage. And so now, after a lot of studies, tree of heaven is often referred to as a highly preferred host. And what we became concerned about in ARS is the potential for them to affect specialty crops. And we were particularly concerned about some of the hosts from Asia that were identified as potential crops at risk, including vitis or grape species, prunus such as peach, and apple or malus, with wine grapes also showing up as being a problem pretty quickly in Pennsylvania, where the insect was first introduced. In terms of tree of heaven or Alanthus altissima, this is an invasive plant as well. It is native to China. It's highly invasive in the continental US, especially the eastern US and California. And according to Plant Bono, I was looking to see if you had tree of heaven on the Hawaiian Islands, and apparently one specimen was found and removed in recent times, but no known additional specimens are present. And hopefully that is still the case because they are highly invasive. But this kind of led to us asking questions really related to the host that this insect could affect, and particularly crops at risk. And so some of the early questions that we asked as part of our research grant program were, what are the specialty crop and common wild host plants bordering specialty crop production areas that would support development and survivorship of this insect? And how could the landscape mosaic that is common in mid-Atlantic, where we have very fragmented environments, contribute to spotted lanternfly pest pressure in vulnerable specialty crops? So this is a tree of heaven with lots of adult lanternflies feeding in September, October. So when we first worked with this insect, we needed to work with it either in a quarantine zone or a quarantine lab. And so our initial work was done in a quarantine lab at Fort Detrick in Frederick, Maryland. And all of this was done in a quarantine greenhouse. And so our first question that we were asking is, what single specialty crop and wild hosts could support their development over a 2-week time frame? So in this case, we were just introducing early instar nymphs, late instar nymphs, the thirds and fourths, and adult into cages with one of these potential hosts. Tree of heaven is a positive control. Black cherry, black walnut, and black locust, which are common woodland species, along with hackberry and sugar maple and white oak, mulberry, as well as apple and peach, some of the crops that are frequently produced in this region. And these trials were conducted over 2 consecutive years in a quarantine greenhouse. So what did we find? Well, what we found was, and this matched some of the field observations that were taken in the early part of its life history, when we have early instars, we saw higher survivorship overall over that 2-week time period, with the greatest survivorship on tree of heaven, followed by black walnut, peach, and sugar maple. Survivorship on all other hosts fell below 50%. That's the red line that you see here at the conclusion of the trial. So 2 weeks on a single host. When we looked at late instars, we saw an even more rapid decline of survivorship below that 50% level, with the greatest survivorship on tree of heaven and black walnut. When we got to the adult stage, the only host that supported survivorship over a 2-week time period as a whole tree plant was tree of heaven. Everything else did not support them very well, with survivorship below 2.5% for all these other hosts. So you can see the importance of tree of heaven with this particular trial. We then wanted to get into some of the specialty crop woody vine hosts that may be at risk, and this is where we started thinking about grape as well as hops and kiwi. And we wanted to do a similar kind of trial with these three host plants, or potential host plants, I should say, compared with Tree of Heaven. So we did the same kind of thing, introducing 50 early instar, 25 late instar, or 10 adults per cage with a single plant, tracking their survivorship and development over a 2-week time period. And in this case, the grape species we were using was Vitis rotundifolia, which the variety is Carlos, and that is a muscadine grape, so different than our wine grapes typically. So in this case, what you can see again, the 50% survivorship line. Survivorship on kiwi fell below pretty rapidly, below 50% at 7 days, whereas the other hosts supported survivorship. But what was interesting was that the fuzziness of the kiwi vine seemed to deter as a physical deterrent for them to be able to feed on the phloem in that tissue. Because if you look at the late instars, you can see significantly greater survivorship on Tree of Heaven, but all of the other hosts seem to do fairly well as well. And then when you get to the adult stage, survivorship on all hosts fell below 50% yet again, except for Tree of Heaven. So they can feed on some of these plants, but again, at the adult stage, it seems like they really need Tree of Heaven. So this is just the percent survivorship of those original hosts that we looked at over the 2-week time period. And so again, the main findings are that Tree of Heaven supports the highest survivorship. Nymphs, early instars can feed on a lot of things and survive reasonably well. Black walnut seems to support high nymphal survivorship, but not so much for the adults. So this kind of led to this next tier of trials that we also did in the quarantine greenhouse, asking how effectively can lanternfly complete their development on specialty crop and wild host plants. In this case, really zeroing in on Tree of Heaven, black walnut, apple and peach, and grape. Again, with the Carlos, the Vitis rotundifolia, as single host diets or as mixed host diets. So in this case, what we did was track their development to the adult stage, and from the time they were first in SARS to the adult stage, so survivorship and development. So they were either given 2 of these single host diets, 2 plants per cage, or a paired host diet. So in this case, what you can see is over time, only Tree of Heaven, again, had the greatest survivorship and provided the greatest level of survivorship and development. But when they were paired with other hosts, this also improved their overall survivorship. And if you look at the overall development over time, so Tree of Heaven alone, survivorship and development to the adult stage was about just a little less than 50%. With these paired hosts, Tree of Heaven with apple, grape, peach, or walnut, they also had high survivorship, but you can see apple alone, they didn't get past third instar. Grapes, the Carlos, the rotundifolia, they never got past fourth instar, and peaches only to the second instar. Black walnut did support development to the adult stage, but it was much lower. And the other thing to know about these mixed diets is that they develop much faster. In fact, it was significantly so. And so you can see that when they had mixed diets, they also survived quite well in the adult stage. So you see these mixed diets as well as Tree of Heaven alone, they were living somewhere between 50 to 60 days, well, a little lower, maybe 40 to 60 days. Whereas if you only provided them with black walnut alone, they didn't survive very well. So what this was telling us is that there was really something interesting going on in terms of the importance of Tree of Heaven in their diets. So let me think. Yeah, so this was the survivorship graph over time. So, and just showing that the survivorship dropped off. Okay. Now, this is the same kind of thing where we can look at combining grape, in this case, Vitis vinifera, or black walnut as a primary host and doing mixed diets. And what you can see in this case, it's the same kind of experiment, but the difference is wine grapes compared with Carlos or the rotundifolia support development and survivorship. They can develop to the adult stage. And when they have mixed diets, we see pretty high survivorship um, for grape plus tree of heaven, I should say Vitis vinifera, as well as black walnut. So these hosts, Vitis vinifera seems to be a very vulnerable crop and also a good host. Black walnut as a wild host, it's not very good on its own or in most mixed diets, except with Tree of Heaven. So while it supports development to the adult stage, it's fairly low and they don't survive long on it um, after completing development to the adult stage. Okay, so the last of these series of experiments that I wanted to show you is just trying to get at the different Vitis species or grape species, um, how effective a host they are. So in this case, we were doing the same kind of thing where we were introducing newly hatched first instar nymphs with Vitis vinifera Chardonnay, Vitis vinifera Pinot Noir, wild species, riverbank, Vitis riparia, Labrusca, which is your typical juice grape that you would find in grape juice in the grocery store, rotundifolia, which we know is not very good, and Tree of Heaven again as a positive control. Again, these were done at Fort Detrick. And what was interesting, again, of course, Tree of Heaven on top in terms of survivorship and um, um, throughout the course of the trial. But you can see that wild grape species, Riparia, Pinot Noir, and Chardonnay also seem to be somewhat improved in terms of overall survivorship and development. And in fact, what we saw was that these other species, or these other cultivars, I should say, of cultivated wine grapes like Pinot Noir, as well as Chardonnay, seem to be pretty good hosts. Riparia, the wild grape vine species known as river grape, also can support populations. So all of this has implications for its survivorship in unmanaged areas. Again, rotundifolia, not a good host. And the juice grapes, while they did make it to the adult stage, you can see we only had a few individuals. So we're really zeroing in on some of our cultivated grape species as being the crops at risk. But we also wanted to convince ourselves that that tree fruit was not going to be affected by this pest because we do see all life stages feeding on them. And so we asked the question, do spotted lanternfly feeding on, does their feeding on young apple and peach trees impact the plants themselves? And so we did this in collaboration with our horticulturist, where we looked at Honeycrisp apples and the Saturn variety of peaches, and we introduced the individuals into these cages season long, so and representing the life stage that was in the field, so early instars through about the middle of July to late instars to adults, and we would replace anybody who died over the course of the week. Then we planted these exposed plants that had been in pots into a field, and we tracked the damage throughout not just this period when we first introduced them into the cages, but the following two field seasons, just to look at how well or how much they may have impacted these plants in their growth. What we found was for apple, we saw absolutely no impact from spotted lanternfly feeding. However, for peaches, we saw when the early instars are feeding on these young peach trees, we did see a reduction in growth for infested plants. But again, they don't survive very well on it, and we were replacing individuals. But what we hadn't expected, but it wasn't a significant effect, was it actually increased frost susceptibility the following season. So those that previously were infested had far more frost injury than those that were not infested. And this kind of aligns with some observations made in Pennsylvania, where wine grapes that had been fed on heavily seem to be more susceptible to cold winter injury. So it seems like when these insects are feeding on the phloem, they're taking out a lot of resources that may provide some ability to deal with abiotic conditions that are adverse, like cold weather or frosts, and it makes them more susceptible. So overall, we're not too worried about apple. We saw some impacts on peach, but again, they don't survive well in peach, so we're hopeful that we won't see any further impacts. We also wanted to ask the question, how acceptable are these key wild and cultivated host plants when we pair them up in the field? So in other words, if you provide them with these plants in the field, will they remain on them and feed? This gets back to what we were saying. Well, we saw these impacts on peach. Would they actually remain there? So what we did was to do a mark release recapture experiment where we paired Tree of Heaven with another host plant, apple, peach, grape, black walnut, or tree of heaven. And we would release marked individuals at the base of each of these plants and track their movement. Would they remain on the host that they were released, or would they move over these different intervals? And we did this for early instars, late instars, and adults. And this we hoped would provide us with a better sense of if these insects would remain on these plants and feed on them under field conditions. So what we found, this is the early instars, the late instars, and the adults. For Tree of Heaven, paired with Tree of Heaven, obviously no significant difference. But when we paired them with Riesling, grape, Vitis vinifera, we also saw no movement. So what that is telling us is that Riesling grape is a very good host and they're quite happy to sit on it and feed. Versus something like apple, where you see the early instars moving to Tree of Heaven, the late instars moving to Tree of Heaven. The adults, we didn't see much movement, but adults typically sometimes just will kind of sit there and not get up to much compared with some of these early instars where they're seeking nutrients. Peach, we had the same kind of pattern where they were leaving the host. And for black walnut, we also saw, even for the adults, they were moving to Tree of Heaven. So all of this information we're using to try to understand host acceptability and host usage under field conditions. The last thing we tried to do to inform ourselves what hosts might be most acceptable under field conditions was to deploy sentinel plants, both wild and cultivated host plants for SLF in the field, and look at numbers of insects on them season long. And what we saw, and this is the graph throughout the season, was that obviously Tree of Heaven, the green line, we had constant SLF on them throughout the season. Black walnut was also very apparent, but we saw in this case fewer individuals, and this was a muscadine grape, so fewer individuals on the apple, peach, and muscadine grape throughout the season. So all of this is telling us that there are certain hosts that they're going to feed on throughout the season, like Tree of Heaven, but other hosts that are vulnerable, like Vitis vinifera, as well as some of the wild grape species like riverbank grape. So our host plant conclusions indicate that Tree of Heaven is a highly acceptable host, as well as Vitis vinifera. Riparia is a good host. Rotundifolia is not. Black walnut is very suitable for nymphal life stages, but not so much for adults. And I don't think we have to worry about apple and peach as crops at risk from this host, but grape, absolutely. But overall, what it tells us is the mid-Atlantic landscape and the host plants within it likely favor establishment and spread. And if you look across our landscape, we have a lot of vineyards in the area, particularly Northern Virginia, where we can see feeding and reproduction in specialty crops like grape vineyards, but we also see them feeding on some of our native and invasive woody hosts outside the specialty crop production areas. And so that tells us that this insect, once it invades a landscape, if it has these kinds of features, this could allow for establishment. And so this gets into the other topic that I wanted to share with you today, and that is some of our work on dispersal and the fact that, excuse me, strong dispersal can be a mechanism for spread, obviously, of invasive species. Now, of course, we have potential for let me have a little sip of soda water. So obviously, we can have human-assisted transport of eggs. Eggs are very they sometimes just look like bits of mud and they may be nondiscrete and people may not know and they move them. But there have been a lot of campaigns for people to check their goods and their materials and outdoor furniture if they're going to move, that kind of thing. And that definitely has helped. But we also have other life stages, the mobile life stages of this insect. And one of the things that we've noted, if you look at this map that I showed you earlier, is that the distribution and the spread of this insect has moved along major interstates north to south, east to west, as well as railroads. And oftentimes these new populations are popping up in urban centers. So we started thinking about the potential for human-assisted dispersal. And, we have some sites near railroad tracks, and we often see individuals kind of hanging around and looking like, oh, they could possibly hitch a ride. And obviously, you saw on my intro slide an insect taking a ride on someone's car. So this led to us to think about this more deeply in the fact that there is a long historical record of human-assisted dispersal in cargo, plant materials, planes, trains, ships. And we know that brown marmorated stink bug, emerald ash borer, spongy moth, Asian longhorn beetle are all examples that humans have just, inadvertently helped move these insects around. So we wanted to empirically determine if accidental hitchhiking by SLF, the mobile life stages, could be an important dispersal mechanism and pathway to spread. And specifically, what we ended up doing is working with our engineering staff to use this laminar flow fan that was powered by a hydraulic motor to develop a vehicle-based human-assisted dispersal bioassay. And so our question was, can we create a system that enables us to understand the impact of hitchhiking or human-mediated dispersal on spotted lanternfly spread? Can we measure the amount of adhesion and dislodgement of potential hitchhikers? So we had these anemometers at different points on the vehicle to measure wind speed. And we looked at initially these 5 locations: the side panel, the window well, the wipers, the windshield, and the hood, because we have different materials like glass and rubber and painted surfaces, as well as orientation. We also wanted to look at all life stages. So we had first, second, third, and fourth instars, as well as early adults, which are kind of pre-reproductive, versus late adults, which are generally actively reproducing. And we would gently place them on the car in terms of acclimated individuals, and we would there was no airflow. And then when they started moving, or about 5 seconds later, we would slowly crank up the wind machine, and it would take 30 seconds to reach maximum wind speed. So that's acclimated. And so, and we would look to see when the insects would become dislodged. Non-acclimated, we wanted to kind of simulate what might happen if an insect just dropped on a car and either it was moving or somebody took off immediately. And so they really were provided no acclimation. They were kind of put on the car and the wind was going. So those were the 2 kind of situations that we tried to simulate. So in terms of the results, what we saw was that if you look at the kilometers per hour for dislodgement, we saw that the early instars and some of the early adults, as well as I should say the first and seconds, as well as the early adults, hung on a bit longer for acclimated, as well as for non-acclimated for the early instars. Then if you look at the different life stages combining the two because we actually saw no significant difference. Again, these violin plots show both the overall spread of the data. And again, first INSARs and early adults came out on top in terms of being able to remain on the vehicles for the longest period of time. And you can see the mean value here with the darker line. In terms of the different locations on the vehicle, this is where it was kind of amazing. What we saw was on some locations, all life stages could hang on to the highest wind speed. And our highest wind speed was about 65 miles per hour. So in the window well, which is a little bit more protected, as well as the wipers, we were seeing all life stages hang on. And even places like the side panel and the windshield, they were able to cling for quite a period of time. And so what this tells us is that even though it may not happen every time, it's the rare events that we worry about with invasive species, that these insects definitely could hitch a ride. So in terms of some of the things that we've noticed with this is that there are kind of postures the insect can take that allows it to be either dislodged or successfully adhere. And so when they're a little bit, if they're kind of more upright where the wind can catch them, they generally are dislodged. Whereas if they have kind of a lower profile, as you see below, they're able to hang on a little bit more. Another thing I should mention is that we did look at survivorship of these dislodged individuals. Would they actually survive once they were blown off? And about 50% of them did survive. So even when they are dislodged, they're not necessarily dying from that, the wind itself. So overall, from this work, what we were able to see is that first INSAR nymphs and early adults were able to adhere. The well and the wiper were certainly the best locations, but again, all life stages were able to reach the maximum wind speed at some locations on the vehicle. And so next steps, we're hoping that some of the modelers at Temple will use some of these to help define areas of spread and identify high-risk areas where they may leave one area and show up as well. So those are the two main research areas that I just wanted to touch on for today. In terms of what we're doing next, I'll just talk about two projects that we have going on. One is managing spotted lanternfly in Vitis vinifera in wine grape vineyards. One of the things that we've been doing is looking at the amount of time it takes from when you find, in this case, one dead spotted lanternfly in your parking lot until you have a damaging population. And so we went from 2019 from one dead spotted lanternfly in our laboratory parking lot to the following year where we were able to pick up positive environmental DNA samples, meaning the DNA left behind by spotted lanternfly on plant surfaces, we were able to capture that, pick it up, and indicate that the lanternfly was present, even though some of our monitoring traps and visual samples were negative. 2 years later, all of these sampling methods yielded positive SLF in them, whether it's visually, trapping, or eDNA. By 2022, we already have very conspicuous populations. And last year, for the first time, we had damaging populations on our wine grapes. So it took about 4 years until we had populations. And that's what we've seen through vineyard surveys as well. So we know we have a vulnerable crop and that we have to manage this insect. So we are working on thresholds for growers to use when they should be treating for this insect. And that includes post-harvest, unfortunately, because they're feeding on the phloem and they can reduce the nutrients going into the roots or after they senesce and possibly making them more vulnerable to winter injury. And then the last thing we have going on is an area-wide project. This is a project funded by USDA ARS, and this is a joint project with Virginia Tech, Rutgers, Penn State, and ARS. And what we're trying to do is actually use biocontrol agents and leverage biocontrol across trophic levels. So where we can control tree of heaven as a weed with a verticillium wilt species, and also use entomopathogens against the lanternfly itself. And then hopefully with those two pieces, we can reduce populations and reduce the threat to wine grapes and hopefully get to the point where we really have a sustainable management strategy. So with that, I just want to thank my lab group and just say that if you ever get a chance to see spotted lanternfly in person, in general, they definitely leave an impression. They're quite dramatic. So I'm going to stop sharing and see if there are any questions. Thank you so much.